Good evening and welcome to Dreamy Academy. Tonight we embark on a journey not across distant galaxies or through the deep strata of Earth, but into the intricate silent universe within each of us. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Tonight, we will explore the quiet, ceaseless vigilance that defines life itself, the immune system. It might seem a complex, even daunting subject filled with unfamiliar names and intricate pathways, but beneath the surface of its complexity lies a profound and ancient wisdom, a story of identity and defense that began billions of years ago and continues without pause in every breath you take. Imagine, if you will, the universe as a vast, shimmering tapestry. Within this tapestry, life emerges, tiny, fragile threads attempting to define themselves against the boundless weave. From the very first stirrings of self-replication, a fundamental question arose: what is me, and what is not me? This isn't just a philosophical query, it's a matter of survival, etched into the very fabric of existence. Every living thing, from the simplest bacterium to the most complex human, must constantly answer this question, moment by moment, across the span of its existence. This is the beautiful paradox of the immune system. It is a system born of boundaries, yet it thrives on connection, it differentiates, yet it unifies. It is an internal symphony of defense, echoing the universe's own tendency to create distinct forms from a shared primordial soup. It is a story of wonder, resilience, and the quiet, persistent pulse of life protecting its own delicate flame. Let us drift now into this hidden world, where molecules whisper secrets and cells dance a timeless ballet of recognition and response, all within the silent sanctuary of your own being. Have you ever considered the silent wisdom held within every cell of your body? A wisdom far older than thought or language. It is the wisdom of distinction, of knowing with absolute certainty what belongs and what does not. This fundamental discernment is not a recent invention of complex life. It is a primal whisper, echoing from the very dawn of cellular existence billions of years ago. Picture, if you will, the early Earth, a world of roiling oceans, volcanic exhalations, and a sky unlike our own. In these ancient waters, the first fragile membranes formed, enclosing a nascent chemistry that could stir, replicate, and respond. Each of these protocells, a tiny island of order in a vast, indifferent sea, faced an immediate existential challenge: how to maintain its integrity, its unique internal environment, against the chaotic external world. The boundary, the shimmering membrane, was its first, most crucial declaration of self. This membrane was more than a simple wall. It was a living frontier, a dynamic interface where the cell met its world. It allowed nutrients to enter, waste to exit, and crucially, it held the cell's precious internal machinery, its proteins, its genetic material, safe from dilution and disruption. This act of enclosure, this creation of an interior distinct from an exterior, was the very first immunological act. It wasn't about fighting invaders yet, but about simply being, about defining an identity in a universe that otherwise sought to blend everything into an undifferentiated whole. As life evolved, this simple boundary became more sophisticated. The cell membrane, a delicate bilayer of fatty molecules, began to bristle with intricate protein structures. These weren't mere decorations, they were the cell's unique molecular signature, its identity tags proclaiming, "I am this cell with these specific functions, part of this organism." Imagine each cell as a tiny self-contained planet, adorned with flags and banners that declare its allegiance and purpose. These surface markers are the language of self-recognition, a quiet conversation happening constantly across the vast cellular landscape of your body. But the world, even at a microscopic scale, is not always benign. Other forms of life arose: bacteria, viruses, fungi, each with its own drive to survive, often at the expense of others. So the simple act of defining self had to evolve into the more active process of recognizing non-self and then, crucially, responding to it. The serene boundary became a vigilant border, and the quiet molecular conversation became a constant watch. Consider a bacterium, a single-celled life form itself, yet profoundly different from the cells of your body. It carries its own unique molecular patterns, distinctive sugars, fats and proteins on its surface that are not found on human cells. These patterns are like foreign flags, alien signatures that immediately signal other. The early immune system, even in its simplest forms, developed the capacity to detect these universal foreign patterns, these conserved molecular motifs that scream, "intruder." This detection wasn't initially about intricate, tailored responses. It was about ancient, generalized alarms. Imagine a simple guard at the city gate not knowing every individual citizen by name, but instantly recognizing anyone not wearing the city's uniform, or perhaps anyone carrying a weapon. Early immune cells, like these guards, developed receptors that could bind to these common non-self patterns. When a receptor encountered such a foreign signature, it triggered a cascade of internal events within the cell, a distress signal, a call to action. These early responses were often direct and decisive. A cell might simply engulf the foreign particle, swallowing it whole in a process called phagocytosis, and then dismantling it within its own internal compartments. Or it might release toxic compounds to neutralize the threat. These are the ancient foundational tools of immunity, honed over billions of years, present in some form in almost all multicellular life. They are the bedrock of what we call innate immunity, the body's immediate non-specific defense system, always ready, always watching. The elegance here lies in its efficiency. No need to learn about every specific threat. Simply recognize the common, unchanging features of invaders, the patterns that signify not one of us. This is a profound evolutionary shortcut, allowing life to defend itself against a vast array of potential dangers with a relatively small set of molecular tools. It's a testament to the universe's tendency toward elegant solutions, where simple rules, applied over deep time, yield astonishing complexity. What this tells us is that the immune system is not just a collection of cells and molecules that fight disease. It is the living embodiment of identity. It is the story of how life learned to distinguish itself, to protect its unique spark, and to maintain its delicate balance against the boundless flux of the world. It is a system that works tirelessly, silently, without your conscious awareness, ensuring that the whisper of self is never drowned out by the clamour of non-self. So we've seen how the very first stirrings of life necessitated a fundamental distinction between self and non-self, giving rise to ancient, elegant mechanisms of defence. This foundational wisdom, etched into the earliest cells, forms the bedrock of all immunity. But how does this primal whisper translate into the complex, coordinated symphony of defense required for a multicellular organism, a vast metropolis of trillions of cells? How do these individual sentinels communicate and cooperate to protect the entire being? That question leads us into the grand architecture of innate immunity, a system of immediate universal vigilance that guards the gates of your existence, which we will explore next. And so we stand at the threshold of a new understanding, gazing upon the intricate canvas of life that has evolved far beyond the solitary, self-defining membrane of a single cell. Chapter 1. The primal whisper of self laid bare the ancient truth. The very essence of existence hinges upon the discernment of self from non-self. But how, you might wonder, does this foundational whisper, born in the primordial soup, amplify into the grand protective chorus of a complex, multicellular organism like you or I? How do these individual sentinels, these tiny self-contained planets, communicate across the vast internal landscapes of our bodies, forming a cohesive, vigilant border against the incessant tide of the unknown? Imagine, if you will, that initial protocell, a solitary pioneer in a boundless ocean. Its membrane, a shimmering declaration of identity, was its sole defense. But as life blossomed, as single cells began to coalesce, to cooperate, to form the magnificent sprawling cities we call tissues and organs, the demands on this primal defense system grew exponentially. No longer could a single cell bear the entire burden of recognition and response. A division of labor was needed, a specialized force, a network of interconnected guardians, each playing a vital role in the preservation of the collective self. This, dear friend, is where the silent symphony truly begins to find its instruments. Within the intricate tapestry of our multicellular existence, the innate immune system has diversified, refined its ancient wisdom, and deployed a remarkable array of specialized cells, each a master of its particular craft, yet all united by that fundamental directive: protect the self. Consider the macrophages, for instance. Their name, meaning big eaters, hints at their primary role. These are the vigilant, roving patrols of your internal universe, the silent sweepers of your tissues. Born from the same lineage as those early phagocytic cells, macrophages are the direct descendants of life's first defenders. They drift through the lymphatic system, patrol the bloodstream, and stand guard in almost every tissue, their surfaces bristling with an array of receptors. These receptors are like sophisticated antennae, constantly scanning for those universal foreign flags, those PAMPs, pathogen-associated molecular patterns, that signify an intruder. When they detect a threat, be it a rogue bacterium or cellular debris, they extend their pseudopods, flowing around the unwanted particle like liquid light, engulfing it and then dismantling it within their powerful enzymatic chambers. It is a timeless act, a fundamental cleansing, a quiet devotion to maintaining order. Then there are the whole neutrophils, the rapid responders, the first wave of defense when an alarm sounds. Imagine them as the elite shock troops of the immune system, constantly circulating in your bloodstream, poised for immediate deployment. When a tissue is breached, when those foreign patterns are detected, neutrophils are among the first to arrive, rushing to the site of infection with a singular, fierce purpose. They too are voracious phagocytes, capable of engulfing vast numbers of pathogens. But their aggression is often short-lived. They are designed for a swift, overwhelming assault, often perishing in the battle, their sacrifice a testament to the system's unwavering commitment. Their lifespan is brief, but their impact is profound, forming a crucial barrier against the initial spread of infection. And what of the cells that watch over our own? The immune system, in its boundless wisdom, knows that sometimes even self cells can turn rogue, compromised by viral infection or cancerous transformation. For this, we have the natural killer cells, or NK cells. These are not phagocytes in the same way. Instead, they are direct assassins, trained to recognize the subtle alterations in the surface of a self cell that indicate distress or danger. They don't look for foreign patterns. Instead, they look for the absence of proper self markers or the presence of stress signals. Like a skilled artisan recognizing a flaw in a masterwork, NK cells identify these compromised cells and induce them to undergo programmed cell death, a gentle altruistic dismantling that prevents the spread of harm to the greater organism. It is a profound act of self-sacrifice ensuring the health of the whole. But how do these diverse sentinels, the patrolling macrophages, the rushing neutrophils, the watchful NK cells, know where to go, what to do, and when to act? This brings us to the very heart of the grand architecture of innate immunity: communication. In a system so vast, so interconnected, a silent language must exist, a series of chemical whispers and shouts that coordinate the defense. This internal language is spoken through a remarkable family of molecules, the cytokines and chemokines. Think of them as the intricate postal service and navigation system of your body's immune network. Cytokines are the alarm bells, the distress signals, the urgent bulletins broadcast across the internal landscape. They are small proteins released by various cells, including those very macrophages and neutrophils we just discussed, when they detect a threat. When a macrophage encounters a bacterial invader, it doesn't just engulf it, it also releases a cascade of specific cytokines. These molecules travel through the bloodstream and interstitial fluids, carrying vital information. Some cytokines act as urgent calls to arms, activating other immune cells, telling them to ramp up their defenses, to proliferate, to become more aggressive. Others act as messengers to distant parts of the body, signaling the brain to induce fever, a deliberate raising of the body's temperature, creating an environment less hospitable for many pathogens. Still others initiate the process of inflammation, a localized storm that is, paradoxically, a profound act of healing. And if cytokines are the general alarm, then chemokines are the precise navigation beacons, the invisible scent trails that guide immune cells to the exact coordinates of trouble. Imagine a fire breaking out in a bustling city. The general alarm, cytokines, alerts everyone, but the specific address, chemokines, directs the fire trucks to the right street, the right building. When tissues are damaged or infected, the local cells release specific chemokines. These molecules create a concentration gradient, a chemical breadcrumb trail that immune cells with their specialized chemokine receptors can follow with remarkable precision. A neutrophil circulating aimlessly in the bloodstream can suddenly detect this gradient and, like a compass needle swinging to true north, begin to move purposefully towards the source of the signal, squeezing through the walls of blood vessels to reach the battleground. This orchestrated response, this symphony of cellular action and molecular communication, culminates in the powerful protective process we call inflammation. Have you ever scraped your knee or felt a splinter embed itself in your finger? The redness, the warmth, the swelling, the slight discomfort, these are not signs of failure, but rather the visible manifestations of your innate immune system at work, a localized storm designed to clear the invaders and initiate repair. When tissue is damaged or pathogens are detected, 1. Local cells release cytokines and chemokines. 2. Blood vessels near the injury dilate, increasing blood flow causing redness and warmth. 3. The walls of these vessels become more permeable, allowing fluid, proteins and immune cells, like those eager neutrophils and macrophages, to leak out into the affected tissue causing swelling. 4. Chemokines guide these immune cells directly into the site of infection. 5. Once there, they engage in phagocytosis, release antimicrobial compounds, and continue to communicate with other cells, amplifying the response. This entire process, this intricate ballet of molecules and cells, is astonishingly rapid and efficient. It is the immediate non-specific line of defense, ready to act against any perceived threat, relying on its ancient wisdom to recognize those universal non-self patterns. It doesn't need to learn about a specific pathogen. It simply recognizes the hallmarks of invasion, the common signatures of danger that have been present since the dawn of life. So the primal whisper of self-defense, once confined to a single membrane, has indeed scaled. It has blossomed into a vast, interconnected network of specialized cells, each a vigilant guardian, communicating through a sophisticated language of chemical signals. This is the silent symphony of innate immunity, playing its timeless melody of protection within you, every moment of every day, without your conscious awareness. It is a testament to the profound resilience of life, an ancient wisdom embodied in every cell, tirelessly working to preserve the unique spark of your identity. As we gently drift further, we begin to appreciate the sheer ingenuity of this foundational defense. But even this grand architecture, as powerful and ancient as it is, has its limits. What happens when a pathogen is particularly cunning, when it evolves to evade these universal detectors, or when the sheer scale of the invasion overwhelms the innate response? How does the body remember past invaders, becoming stronger and more precise with each encounter? These are the questions that beckon us deeper into the intricate, silent universe within. And so we arrive at a pivotal question, don't we? What happens when the ancient universal wisdom of our innate defenses encounters an adversary it cannot immediately decipher? What if the common molecular patterns, the foreign flags our macrophages and neutrophils are so adept at spotting, are subtly altered, or entirely new? What if the invader, cunning and adaptable, evades the initial sweep, or simply overwhelms the front line with sheer numbers? You see, the universe within us, vast and intricate, is never content with a single layer of defense. It holds reserves, a deeper wellspring of intelligence poised to awaken when the primal whisper of innate immunity requires a more sophisticated, more tailored response. This, my dear friends, is where we journey into the realm of adaptive immunity, a system of astonishing specificity, profound memory, and a dynamic capacity for learning. Imagine, if you will, that your innate immune system is a vigilant border patrol scanning for known threats with general descriptors. Anyone carrying a weapon? Anyone without proper identification? It's effective, immediate, and crucial, but the adaptive immune system, that's the highly specialized intelligence agency, the master code breakers, the elite snipers, and the archivists who remember every face, every fingerprint, every nuanced detail of every past encounter. It learns, it remembers, and it adapts. This magnificent capacity for refined recognition begins with an idea, a principle that is both simple and breathtakingly complex: the antigen. Where innate immunity recognizes broad shared patterns, adaptive immunity zeroes in on unique molecular signatures. Think of antigens as the precise individual fingerprints of every potential threat. A specific protein on a virus's coat, a unique sugar chain on a bacterial wall, even a subtly mutated protein on a cancerous self-cell. These are the markers that declare, unequivocally, I am this specific entity and no other. And how does our body learn to recognize these myriad distinct antigens? It possesses a dedicated library, a vast and quiet repository of potential keys, ready to be matched to any intruder's lock. This library is housed within two extraordinary types of white blood cells, the maestros of adaptive immunity, the Sao B lymphocytes, or B cells, and the SAS T lymphocytes, or Paldo T cells. Let us first turn our gaze to the B cells, these elegant architects of humoral immunity. Imagine each B cell as a tiny self-contained planet, its surface adorned with thousands of unique molecular antennae, specific receptors, each one a potential key designed to fit a particular antigen lock. The sheer diversity is mind-boggling. Our bodies possess B cells capable of recognizing an almost infinite array of antigens, even those that have never before existed on Earth. How is this possible? Through a miraculous process of genetic rearrangement, a biological lottery that shuffles and recombines gene segments to create an astonishing diversity of these receptor keys, long before an invader ever appears. So I'm on a bia or a mia. Cell drifts through the lymphatic system, a silent centered, waiting. When its specific receptor encounters and binds perfectly to its matching antigen, that unique fingerprint of a pathogen, it's like a spark igniting. This binding is often the first signal, but for a truly robust response, the B cell usually needs a co-pilot, a crucial confirmation from a helper T cell, which we will meet shortly. Once fully activated, this B cell undergoes a profound transformation. It begins to divide rapidly and exponentially, creating an army of identical copies, a process known as clonal expansion. But these aren't just more B cells. Most of them evolve into powerhouse factories called plasma cells. Picture a plasma cell as a dedicated artisan, no longer displaying its receptor keys on its surface, but instead furiously manufacturing and secreting millions upon millions of soluble versions of that exact key. These secreted keys are what we call antibodies. Antibodies are the true marvels of humoral immunity. They are Y-shaped proteins, exquisitely designed to circulate throughout our blood and lymphatic fluid, seeking out and binding to the very antigen that triggered their creation. Think of them as guided missiles launched with pinpoint accuracy. When an antibody binds to its target antigen on a pathogen, it doesn't directly destroy it. Instead, it acts as a signal, a flag, a tag, marking the invader for destruction by other immune cells. An antibody might coat a bacterium, preventing it from attaching to our cells. It might neutralize a virus, blocking its entry into our cellular fortresses. It might clump together many pathogens, making them an easier meal for macrophages to engulf. Or it might activate the complement system, a cascade of proteins that can directly puncture and lyse bacterial membranes. In essence, antibodies are the bridge between the specific recognition of the adaptive system and the brute force efficiency of the innate system, guiding the ancient defenses to precisely where they are needed most. Now, let us turn our attention to the T cells, the other formidable architects of adaptive immunity, responsible for what we call cell-mediated immunity. Unlike B cells, which can directly see antigens floating freely, T cells are more discerning. They don't recognize raw antigens. Instead, they require antigens to be presented to them, much like a detective presenting evidence in court. This presentation occurs on the surface of other cells, specifically on specialized molecules called major histocompatibility complex (MHC) proteins. Imagine MHC proteins as display pedestals, present on almost every cell in your body. When a cell becomes infected, or when a macrophage engulfs a pathogen, it breaks down pieces of the invader into small peptide fragments, the antigens. These fragments are then loaded onto the MHC pedestals and displayed on the cell's surface. It's a universal "look what I found" signal, or in the case of an infected cell, a desperate "I'm compromised" plea. T cells too possess unique receptors on their surface, but these receptors are designed to recognize not just the antigen, but the antigen in the context of an MHC molecule. It's a two-part lock and key. We have two main types of T cells, each with a distinct and vital role. First, meet the helper T cells, often called CD4+ T cells. These are the master coordinators, the strategists, the indispensable conductors of the immune symphony. When a helper T cell's receptor binds to an antigen presented on an MHC molecule by an antigen-presenting cell like a macrophage, it becomes activated. Once activated, helper T cells begin to release a torrent of signaling molecules, those familiar cytokines we discussed earlier. These cytokines act as powerful stimulants, shouting commands to other immune cells. They are the essential activators for B cells to fully transform into plasma cells, for cytotoxic T cells to become fully lethal, and for macrophages to become even more voracious. Without helper T cells, the adaptive immune response would falter, like an orchestra without its conductor. Then we have the formidable cytotoxic T cells, often called CD8+ T cells, the elite assassins of the immune system. Their mission is chillingly precise: to seek out and destroy our own body cells that have been compromised, typically by viral infection or cancerous transformation. When a cytotoxic T cell recognizes an antigen displayed on an MHC molecule on an infected or cancerous cell, a cell gone rogue, it latches on. With deadly efficiency, it then delivers a lethal kiss, injecting toxic granules that induce the target cell to undergo programmed cell death or apoptosis. This prevents the spread of viruses and eliminates potential cancer threats, all while minimizing collateral damage to healthy tissue. It's a silent, necessary sacrifice for the greater good of the organism. Just like with B cells, when a specific T cell, whether helper or cytotoxic, encounters its matching antigen presented on an MHC molecule, it undergoes clonal expansion. The few rare T cells capable of recognizing that specific threat multiply into a vast army, all perfectly equipped to combat the invader. It's an amplification of the perfect response, ensuring that enough specialized warriors are on hand to clear the infection. But perhaps the most extraordinary aspect of adaptive immunity, the true hallmark of its wisdom, is its capacity for immunological memory. Once the immediate threat is neutralized and the battle won, most of the expanded effector B and T cells eventually perish. However, a small, highly specialized population persists. These are the memory B cells and memory T cells. Imagine these memory cells as seasoned veterans, retired from the battlefield but forever on standby. They are more numerous than their naive counterparts, and crucially, they are far more easily activated. Should you ever encounter the same specific pathogen again, these memory cells are poised to spring into action with unprecedented speed and power. The response is swifter, stronger, and often so effective that you never even realize you've been reinfected. This is the very foundation of vaccination, a testament to the body's profound ability to learn from experience and prepare for the future. So you see, the innate and adaptive immune systems are not two separate entities operating in isolation. They are deeply intertwined, a seamless continuum of defense. The innate system, with its immediate broad-spectrum response, acts as the first line of defense, buying precious time. It also plays a vital role in presenting antigens to the adaptive system, providing the crucial signals that awaken the specific B and T cells. In turn, the adaptive system, with its exquisite specificity and enduring memory, refines the attack, precisely targets the invader, and ensures long-term protection. Together they form a silent symphony, a vigilant border, a living embodiment of identity, constantly distinguishing me from not me, protecting the delicate flame of life with ancient wisdom and astonishing complexity. As you drift deeper into this understanding, perhaps you feel a quiet gratitude for this intricate, silent universe within you, tirelessly working, learning and remembering, ensuring your peace and well-being. And as we continue our journey, we will explore even more wonders of this incredible system, uncovering layers of defense you never knew existed. For now, rest in the knowledge of this profound internal wisdom. As we drift deeper into the quiet sanctuary of the Dreamy Academy, you've begun to understand the two magnificent pillars of your immune defense, the ancient immediate wisdom of innate immunity and the sophisticated learning prowess of adaptive immunity. We've seen how the vigilant patrols of macrophages and the rapid deployment of neutrophils stand guard, how the precise assassins of NK cells maintain order, and how the adaptive forces of B cells and T cells craft bespoke defenses, remembering every encounter. But here, in the hushed chambers of deeper understanding, we uncover a profound truth. These are not separate armies, acting in isolation. No, they are threads in a single shimmering tapestry, each colour and texture interwoven with the next, creating a fabric of protection that is far stronger, far more intricate than the sum of its parts. They are partners in an eternal silent dance, a seamless collaboration that defines your very existence. Have you ever considered the invisible bridges that connect these worlds? The crucial conduits of information that allow the primal scream of innate danger to awaken the precise, strategic response of the adaptive mind. This is where our journey takes us next, to the heart of this astonishing integration, revealing how your body orchestrates its grand defense. Imagine, if you will, tiny specialized cells acting as sophisticated messengers, bridges spanning the chasm between immediate recognition and learned response. We call them antigen-presenting cells, or APCs, and among them, the dendritic cells are the most masterful. These remarkable entities are born from the innate system, yet their most vital role is to serve as educators for the adaptive one. Picture a dendritic cell as a diligent scout, its many branching arms, like the roots of an ancient tree, constantly reaching out, sampling the environment in your peripheral tissues. It patrols the skin, the lining of your lungs, your gut, every potential entry point for a foreign invader. When it encounters something amiss, a bacterial fragment, a viral particle, a piece of a damaged cell, it doesn't just engulf it like a macrophage might. No, it captures these molecular fragments, these foreign flags we call antigens, and begins a profound transformation. This initial encounter is the primal whisper, the recognition of non-self by the innate system. But the dendritic cell doesn't stop there. Once it has seized its prize, it undergoes a journey. It matures, shedding its patrolling guise, and begins a deliberate migration. It travels through the lymphatic vessels, those silent rivers flowing beneath your skin, heading towards the nearest lymph node. Think of the lymph nodes as bustling academies, specialized training grounds where naive T cells and B cells reside. patiently awaiting their call to action. And it is here, in these quiet strategic hubs, that the dendritic cell becomes the ultimate teacher. With the captured antigen proudly displayed on its surface, nestled within its MHC proteins, the very same molecular pedestals we discussed earlier, it seeks out a specific unsuspecting core T cell. This is no random encounter. Each T cell possesses a unique receptor, a molecular key designed to fit only one specific antigen lock. The dendritic cell, with its array of presented antigens, patiently shows its cargo to countless T cells until it finds the perfect match. When a naive T cell's receptor precisely recognizes the antigen presented by the dendritic cell, it's like a spark igniting in the darkness. But a spark alone is not enough to unleash the full power of the adaptive immune system. This is where the wisdom of the integrated system truly shines. The dendritic cell, having sensed danger through its innate pattern recognition receptors, doesn't just present the antigen. It also provides crucial co-stimulatory signals. Imagine these as a secret handshake, a confirmation code. Without this second signal, the T cell might recognize the antigen, but it would not fully activate. Instead, it might even become anergic, unresponsive, essentially learning to ignore that antigen in the future. This elegant mechanism is a safeguard, preventing your adaptive system from mistakenly attacking your own healthy tissues. It's the innate system, through the dendritic cell, verifying that this is a true threat, not a harmless encounter or a misidentification of self. Once these two signals are received, the specific antigen presentation and the co-stimulatory handshake, the T cell springs to life. It begins to divide rapidly, creating an army of identical clones, each programmed to recognize and respond to that specific invader. And among these activated T cells, the helper T cells, CD4, emerge as the true orchestrators, the conductors of this intricate symphony of defense. We touched upon helper T cells in our last session, noting their role in activating B cells and cytotoxic T cells. But their influence is far more nuanced, far more strategic. Depending on the specific signals they receive from the dendritic cell and the type of pathogen that triggered the initial alarm, helper T cells can differentiate into various subsets, each directing a unique facet of the immune response. Consider the complexity. If the threat is an intracellular pathogen, like a virus hiding within your cells, the helper T cells might differentiate into a one phenotype, releasing cytokines that empower cytotoxic T cells and macrophages to hunt down and destroy infected cells. If the threat is an extracellular bacterium or parasite, they might become or cells, secreting signals that promote antibody production by B cells and rally other innate immune cells, like eosinophils, to the fight. There are even 17 cells, crucial for fighting fungal and certain bacterial infections, and the vital regulatory T cells, TREGs, which we will return to shortly. This incredible plasticity of helper T cells ensures that your body mounts the most appropriate and most effective defense for each specific type of threat. It's like a master strategist, receiving intelligence from the scouts, dendritic cells, and then deploying the precise units and tactics required for victory. The innate system hands off the intelligence, and the adaptive system, guided by helper T cells, crafts the perfect counter-strategy, and the communication flows both ways. The adaptive system, once activated, also sends signals back to enhance and refine the innate response. Antibodies produced by B cells can opsonize pathogens, coating them and making them irresistible targets for macrophages to engulf. T cells can release cytokines that supercharge the killing abilities of macrophages, turning them into even more potent cellular warriors. It's a continuous dynamic dialogue, a feedback loop of protection. But what happens when the battle is won? Or more importantly, what prevents this powerful self-replicating defense system from turning its formidable arsenal against your own precious self? This is where the profound wisdom of the immune system truly reveals its depth in its capacity for regulation and tolerance. Among the various helper T-cell subsets, we find the quiet guardians, the peacekeepers of the immune system, the hit regulatory T-cells, TREGs. These remarkable cells are like the wise elders in our dreamy academy, ensuring harmony and preventing chaos. Their primary role is to suppress the activity of other immune cells, to dampen down an immune response once the threat is neutralized, or, crucially, to prevent an attack on your own body. Imagine the immune system as a finely tuned instrument. While the effector T cells and B cells are the powerful notes of defense, the TREGs are the delicate dampeners, preventing the sound from becoming an overwhelming destructive roar. They are crucial for maintaining self-tolerance. The profound ability of your immune system to distinguish between me and not me, and to leave me unharmed. When this delicate balance is disrupted, when TREGs falter, the immune system can mistakenly target self-antigens, leading to the complex challenges of autoimmune diseases. So you see, the immune system is not a collection of independent specialists. It is a grand, integrated network, a living, breathing testament to billions of years of evolution. From the primal whisper of self-recognition at the cellular membrane to the sophisticated, orchestrated dance of dendritic cells, T cells and B cells, every component works in concert. The immediate broad-spectrum response of innate immunity provides the first line of defense and the crucial intelligence. The precise, adaptable, and memory-driven adaptive immunity then refines the attack and remembers the foe, and the regulatory mechanisms ensure that this immense power is wielded with precision and wisdom, always in service of your continued well-being. As you rest here, surrounded by the quiet hum of this intricate biological machinery, reflect on this astonishing collaboration. The silent symphony playing within you is a constant reminder of life's resilience, its ancient wisdom, and its breathtaking complexity. It is a tireless guardian, working without your conscious thoughts, defining you in every molecular interaction, protecting your unique identity against an ever-changing world. We have begun to unravel the core mysteries of this inner universe, but there are still deeper layers to explore, further wonders to behold in the quiet, ceaseless work of your body's most devoted protector. For now, let this understanding settle within you, a peaceful knowing of the incredible world that thrives unseen within. We've journeyed deep into the inner sanctum, haven't we? Through the vibrant, bustling landscape of your own being, tracing the intricate dance of recognition and defense. We've witnessed the tireless vigilance of innate immunity, the profound wisdom of adaptive responses, and the crucial quiet work of regulatory T cells, ensuring that this immense power is wielded with precision, safeguarding the very self it pledges to protect. The system in its ideal state is a masterpiece of balance, a silent symphony where every instrument plays its part in perfect harmony, recognizing the subtle vital distinction between you and not you. But what happens, my friend, when this exquisite balance falters? What if in the vast, complex tapestry of molecular patterns and cellular signals, a thread unravels? What if the compass that guides your immune system, usually so true, begins to waver, mistaking the familiar for the foreign, the cherished self for a dangerous non-self? This is where we encounter the profound paradox of protection, the shadowed side of a system designed for ultimate defense. Imagine, if you will, a magnificent fortress built over eons to repel every conceivable invasion. Its walls are strong, its sentinels unyielding. But one day, the very guards within the fortress walls, the valiant defenders, begin to perceive a part of the castle itself, a tower, a courtyard, a vital chamber, as an enemy outpost. This, in essence, is the heartbreaking reality of autoimmunity. It is a civil war waged within the silent sanctuary of your own body, a profound betrayal where the immune system, once your staunch protector, turns its formidable arsenal against you. This breakdown in self-tolerance, this tragic misidentification, is one of the most perplexing and challenging aspects of immunology. We spoke of central tolerance, where developing immune cells in the thymus and bone marrow are rigorously screened, and peripheral tolerance maintained by the watchful eye of regulatory T cells. But sometimes, despite these layers of protection, the system errs. A T cell or a B cell meant to be silenced or eliminated for recognizing self-antigens slips through the net. It awakens not to a foreign threat, but to a part of your own body perceiving it as an invader. The causes of this profound misdirection are often a complex whisper woven from many threads. There might be a genetic predisposition, a subtle inherited code that makes certain individuals more susceptible, a predisposition that lies dormant until awakened. Think of it as a particular combination lock waiting for the right sequence of numbers to be entered. Then some environmental triggers can play their part. A viral infection that leaves behind molecular echoes resembling parts of your own cells, a bacterial protein that mimics a self-protein, a phenomenon we call for molecular mimicry, or even certain toxins. These external factors can sometimes provide the spark that ignites the internal conflict, tipping the delicate balance. Consider the diverse battlegrounds where this internal strife can manifest. In rheumatoid arthritis, the immune system mistakenly targets the synovial membranes lining your joints, those smooth gliding surfaces that allow for the graceful movement. It perceives them as foreign, mounting an inflammatory attack that leads to pain, swelling, and eventually damage. It's as if the very hinges of your body are under siege, slowly succumbing to the relentless assault of their own protectors. In type 1 diabetes, the immune system tragically turns its attention to the insulin-producing beta cells in the pancreas. These are the vital architects of metabolic harmony, the cells responsible for regulating blood sugar. The immune system, mistaking them for invaders, systematically destroys them, leaving the body unable to produce this essential hormone. It's a silent, devastating attack on a crucial factory within your metabolic landscape. And then there's multiple sclerosis, where the immune system targets the myelin sheath, the protective insulation around nerve fibers in the brain and spinal cord. Imagine the intricate wiring of a vast, complex electrical system. Myelin is the insulation that allows signals to travel swiftly and efficiently. When the immune system attacks it, these vital signals become disrupted, leading to a wide array of neurological symptoms. It's as if the very communication lines of your internal universe are being systematically frayed and cut. These are but a few examples of the immense diversity and profound impact of autoimmune diseases, each a testament to the immune system's formidable power and the tragic consequences when that power is misdirected. It reminds us of the constant quiet vigilance required to maintain the sanctity of self. Yet the immune system can also err in another way, not by attacking the self, but by overreacting to something utterly harmless. This brings us to the realm of hypersensitivity reactions, more commonly known as allergies. Here, the immune system isn't confused about self, it's simply too zealous in its defense against non-self entities that pose no real threat. Have you ever considered the invisible pollen dust that dances on the breeze? The microscopic dander from a beloved pet? Or the proteins in certain foods? To most, these are innocuous, part of the fabric of our environment. But for someone with an allergy, these benign substances, which we call allergens, are perceived as dangerous invaders, triggering an immune response that is disproportionate and at times profoundly uncomfortable or even life-threatening. This is often seen in one type, a hypersensitivity, the immediate allergic reaction. Here, a specific type of antibody, IgE, plays a central, if sometimes problematic, role. Upon initial exposure to an allergen, your B cells produce IgE antibodies specific to that allergen. These IgE molecules then bind to specialized immune cells, primarily Golgi mast cells, which are strategically positioned in tissues like your skin, airways, and digestive tract, the very interfaces with the external world. Think of these mast cells, now coated with IgE, as highly sensitive tripwires, primed and ready. When you encounter that allergen again, it binds directly to the IgE antibodies on the surface of these mast cells. This binding acts like the pulling of a trigger, causing the mast cells to rapidly degranulate, releasing a cascade of potent chemical mediators, most notably histamine. Histamine is a powerful inflammatory agent responsible for the familiar symptoms of an allergic reaction. The sudden swelling, the itchy eyes, the runny nose, the constricted airways, the hives. It's a full-blown emergency response to a false alarm, a frantic ringing of the fire bells when there's no actual fire, only a wisp of smoke from a distant harmless campfire. The irony is profound. The very mechanisms designed to protect us from parasites and dangerous pathogens are, in the case of allergies, deployed against a harmless peanut or a fragrant flower. It highlights the inherent complexity and the occasional overzealous nature of a system built to err on the side of caution. Better, perhaps, to react too strongly to a harmless pollen grain than not strongly enough to a deadly virus. So as we drift deeper into this understanding, we see that the immune system, for all its astonishing complexity and ancient wisdom, is not infallible. It is a living, breathing entity, capable of incredible feats of protection, but also susceptible to miscalculation and overreaction. These challenges, autoimmunity and hypersensitivity, are the shadow side of its immense power, a poignant reminder of the delicate balance it constantly strives to maintain. Yet even in contemplating these vulnerabilities, our wonder only deepens. For the immune system is not static, it is a dynamic, evolving network, constantly learning, adapting, and striving for equilibrium. It is a testament to the ongoing story of life, forever seeking to define and defend its precious self in a world brimming with non-self. As you continue to breathe softly, allowing these intricate concepts to settle into the quiet chambers of your mind, remember the profound wisdom embedded within you. Remember the constant silent work of this extraordinary system, a tireless guardian, ever striving for that perfect delicate balance between protection and peace. And know that even in its occasional imperfections, there is a deep, resonant lesson in the continuous, miraculous unfolding of life. The deeper layers of this intricate system and the remarkable ways we learn to work with it continue to reveal themselves like stars emerging in the darkening sky. We have journeyed through the intricate landscapes of defense, marveling at the immune system's astonishing capacity for recognition, its swift and precise responses, and even its moments of misdirection, the civil wars of autoimmunity, the overreactions of allergy. We have seen how the vigilant guardian, in its earnestness to protect, can sometimes turn its formidable power inward or react with disproportionate force to a harmless whisper of the outside world. But now, as the gentle currents of our exploration carry us deeper, we encounter a different challenge. What happens when the fortress walls, once so robust, begin to crumble? What if the silent symphony of defense, instead of playing too loudly or off-key, suddenly quiets, its instruments falling seers at conductors losing their way? Tonight we turn our gaze to the unseen shadows, to the profound vulnerability that arises when the immune system falters, when its strength wanes, leaving the delicate flame of life exposed. We delve into the quiet struggles of immunodeficiency and the ultimate betrayal, when self itself turns rogue and the very cells meant to belong become a foreign menace, giving rise to the enigma of cancer. Imagine, if you will, the body as a vibrant, bustling city, constantly under the protection of its dedicated immune forces. We have seen these forces in action, repelling invaders, repairing damage, maintaining order. But what if, deep within the command center, a vital blueprint is missing? What if the training academies for the cellular defenders are incomplete or the raw materials for their weapons are scarce? This is the world of immunodeficiency, where the immune system, for various reasons, is simply not strong enough to perform its sacred duty. Sometimes this weakness is a whisper from our very beginnings, a genetic blueprint drawn with a subtle flaw. This is a primary immunodeficiency, a condition woven into the fabric of a person's being from birth. Perhaps a crucial type of B cell never fully matures, leaving the body unable to craft those precise, Y-shaped antibodies that neutralize threats. Or perhaps the T cells, the master coordinators and elite assassins, are unable to develop, leaving the entire adaptive response in disarray. Think of a fortress built with gaps in its walls, or a symphony where the entire string section is missing. Life becomes an incredibly fragile dance, where even the most common, seemingly innocuous microbes can pose a profound, life-threatening danger. The world outside, which most of us navigate with barely a thought, becomes a landscape fraught with unseen perils, a constant silent battle for survival. More often, however, the shadows of immunodeficiency fall later in life, an acquired vulnerability. This is cell's secondary immunodeficiency, a weakening brought on by external forces or internal stresses. It can be a consequence of severe malnutrition, stripping the body of the fundamental building blocks it needs to forge its defenses. It can arise from certain medications, designed to quell an overactive immune response in conditions like autoimmunity or after organ transplantation, but with the inevitable side effect of broadly dampening the body's protective shield. It can be the toll of chronic stress, which subtly erodes the immune system's resilience over time, or the devastating impact of chemotherapy, which, in its vital mission to eradicate rapidly dividing cancer cells, often takes a heavy toll on the equally rapidly dividing immune cells. Perhaps the most profound and widely known example of secondary immunodeficiency is the relentless assault of the human immunodeficiency virus, or HIV. This cunning adversary does not merely attack immune cells, it specifically targets the very architects and coordinators of the adaptive response, the helper T cells. Remember these master conductors, these vital messengers that orchestrate the entire adaptive symphony, guiding B cells to produce antibodies and marshalling cytotoxic T cells to destroy infected cells. HIV systematically dismantles this central command. It infiltrates these crucial cells, turning them into factories for new viruses, eventually destroying them. As the number of helper T cells dwindles, the body's ability to mount a coordinated specific defense against any new threat slowly collapses. The grand symphony of adaptive immunity first falters, then falls silent. The once vigilant city finds its communication lines severed, its generals incapacitated, leaving it vulnerable to invaders it once easily repelled. This profound loss of specific immunity leaves the body exposed to opportunistic infections and, as we will soon see, to another insidious internal threat. For the immune system, the ultimate betrayal comes not from an external invader, but from within. It is the chilling transformation of a self-cell, once a loyal citizen of the body, into a rogue entity, a lawless clone that grows without restraint, stealing resources and threatening the very harmony of the organism. This is the profound enigma of the cancer. Have you ever considered the silent wisdom of immune surveillance? Long before a lump is felt or a symptom appears, your immune system is often already at work, tirelessly patrolling for signs of cellular rebellion. Every day within your body, cells undergo mutations. Most are harmless, some are repaired, but a few can lead to the uncontrolled growth characteristic of cancer. Your immune system, however, possesses an innate ability to recognize these altered self-cells. It sees the subtle molecular changes on their surface, the slight shifts in their protein expressions, the foreign flags they inadvertently hoist. These are not truly non-self in the traditional sense, but a distorted echo of self, a grotesque parody. Here the vigilant, natural killer, NK cells play a critical role, just as they do with virally infected cells. They look for cells that have lost their normal self-markers, the MHC class I molecules that signal, "I am healthy and belong here." Cancer cells, in their desperate attempt to evade detection, sometimes downregulate these markers, inadvertently making themselves targets for NK cells. And the cytotoxic T cells, those elite assassins, are also on constant patrol, guided by helper T cells, ready to identify and eliminate cancer cells that present abnormal antigens on their MHC class I molecules. It's like a cosmic gardener, constantly pruning diseased branches to maintain the health of the entire plant. This concept of immune surveillance suggests that many nascent cancers are detected and eliminated by our immune system before they ever become a clinically significant disease. It is a testament to the immune system's ancient wisdom, its profound understanding of what constitutes a healthy self, and its tireless commitment to maintaining that integrity. Yet, cancer is a formidable adversary, a master of disguise and evasion. The very evolutionary pressures that drive its growth also select for cells that are adept at escaping immune detection. These rogue cells are not passive targets. They are cunning, adapting, and learning to outsmart their internal guardians. Imagine a chameleon, changing its colours to blend seamlessly into its surroundings. Some cancer cells learn to downregulate their MHC class I molecules, effectively becoming invisible to cytotoxic T cells. They shed their unique flags, slipping into the shadows, making it incredibly difficult for the immune system's specific forces to recognize them as altered self. Other cancer cells develop more insidious strategies. They learn to actively suppress the immune response in their immediate vicinity. They secrete powerful immunosuppressive factors, like a fog that descends upon the battlefield, disorienting and paralyzing the immune cells that dare to approach. They might recruit certain types of immune cells, like some macrophages or regulatory T cells, and reprogram them to become their unwitting allies, turning them into agents of immune suppression rather than defense. It's like a cunning general who not only hides his troops but also whispers lies to the enemy's soldiers, turning them against their own command. Perhaps most remarkably, cancer cells can exploit the very immune checkpoints that the body uses to maintain self-tolerance and prevent autoimmune attacks. Remember the brakes on the immune system, the molecular signals that tell an activated T cell to stand down once its job is done, or to ignore a self-antigen. Cancer cells learn to express ligands that bind to these off switches on T cells, essentially pressing the brakes on the immune response, paralyzing the very cells meant to destroy them. It's a profound manipulation, turning the immune system's own safeguards against it, a silent victory through strategic sabotage. So, as we contemplate the vastness of the immune system's challenges, we see a spectrum of vulnerabilities. From the genetic whispers of primary immunodeficiency, through the acquired shadows cast by disease or treatment, to the ultimate internal betrayal of cancer, the immune system faces battles on every front. It is a ceaseless struggle, a testament to the fragility of life and the immense power of our body's silent guardians. Yet even in the face of such formidable adversaries, there is profound beauty in the immune system's tireless effort, its ancient wisdom, and its relentless commitment to defining and defending the self. It continues its work day in and day out in the quiet depths of our being, a constant reminder of the astonishing complexity that allows us to simply be. As we drift deeper into this understanding, we carry with us a renewed sense of wonder and gratitude for this intricate, silent universe within. The last time we drifted through these intricate pathways, we faced the profound betrayal within, cancer. We witnessed its cunning, its ability to morph, to whisper false peace to the very sentinels designed to unmask it. We saw how it could cloak itself in invisibility, how it could disarm the vigilant T cells, turning their formidable weapons against themselves. It was a sobering testament to the limits of even the most ancient and sophisticated defense system, a demonstration that even the most perfect defense can be outmaneuvered by an enemy born from within. But what if, my dear explorer, we could learn to whisper back? What if we could amplify the faint cries of distress, sharpen the blunted blade, or remove the shackles from our body's most dedicated guardians? This, in essence, is the audacious promise of immunotherapy. A new chapter in our shared journey, where instead of merely attacking the invader, we empower the inner defender. For centuries, the fight against disease was a direct assault. Poison the pathogen, excise the tumour, bombard the rogue cells. It was a necessary, often brutal war. Yet with immunotherapy, we embark on a different kind of strategy, a more subtle dance. We are no longer simply generals commanding an external army. We are strategists, working in concert with the body's intrinsic wisdom, learning to speak its language, to retune its silent symphony. We are learning to guide the immune hand, to reorient its gaze, to awaken the sentinel that has been lulled into a dangerous slumber. Imagine, if you will, the immune system as a vast, complex orchestra, each cell an instrument, each cytokine a note. Sometimes in the chaotic performance of cancer, certain instruments fall silent, or the conductor loses its way, its baton no longer directing the vital movements. Immunotherapy, then, is our attempt to restore harmony, to bring the missing instruments back into play, or to help the conductor regain control, to hear the discordant notes and correct them. One of the most profound breakthroughs in this intricate field has been the understanding of immune checkpoints. Have you ever considered the delicate balance required for our immune system to function? It must be powerful enough to eradicate threats, yet restrained enough not to turn upon its own precious self. This balance is maintained by a series of breaks or checkpoints, molecular switches that tell immune cells, particularly T cells, when to stand down. Think of them as vital safety mechanisms, preventing a full-scale, uncontrolled assault that could lead to widespread self-destruction, a safeguard against the very autoimmunity we explored earlier. These checkpoints, like the PD-1 PDL1's pathway or CTLA4, are essential for maintaining self-tolerance, ensuring our T cells don't mistakenly attack healthy tissues. They are the quiet whispers of all clear, the gentle hand on the shoulder saying, "Stand down, friend. This is one of ours." They are the necessary pauses in the vigilant watch, moments of calm designed to conserve energy and prevent friendly fire. But here's where cancer, in its chilling brilliance, exploits this very wisdom. It learns to mimic these signals. Rogue cells, instead of waving a foreign flag, learn to display the very molecular patterns that activate these immune checkpoints. They essentially pull the emergency brake on our T cells, creating a zone of immune privilege around themselves. The T cell poised for attack receives the stand-down signal, its activation halted, its mission aborted. The cancer thrives, undisturbed, right under the nose of its would-be destroyer, cloaked in a veil of false recognition. This is where checkpoint inhibitors enter the stage. These remarkable molecules are designed to block these deceptive signals. They are like removing the bean folds from our T cells, or perhaps more accurately, unjamming the off switch that cancer has deliberately pressed. By blocking PD-1 on the T cell, or PDL1 on the cancer cell, or CTLA4 on the T cell, we effectively release the brakes. We allow the T cell, previously silenced and paralyzed, to reawaken, to recognize the true nature of the rogue cell, and to unleash its full cytotoxic power. It's like freeing a powerful guardian from an invisible cage, or lifting the spell of confusion that had rendered it inert. The T cell, once confused and disarmed, now sees the tumor for what it is, a foreign entity, a threat to the harmony of the self, and with that recognition, the ancient primal directive to protect is reactivated. We are not introducing a new weapon, you see. We are simply allowing the existing, perfectly designed weapon to do its job. It is a profound act of re-empowerment, a subtle nudge that realigns the compass of defense. Yet the immune system is a vast and varied landscape, and sometimes simply removing the brakes isn't enough. Sometimes the T cells themselves need a more direct, a more tailored instruction. Sometimes the enemy's disguise is so complete, or its signals so muted, that the T cell needs a new pair of eyes, a custom-built sensor. This brings us to another astonishing frontier: CAR T-cell therapy. Imagine for a moment that our T cells are highly skilled, specialized detectives trained to recognize specific molecular fingerprints. But what if the cancer has developed a disguise that is just a little too subtle for even the best detective? What if we could give our detectives a brand new, highly sensitive, custom-designed sensor specifically tuned to one unique feature of the criminal? That, in essence, is what CAR T-cell therapy achieves. We begin by gently extracting a sample of your own T cells from your blood. These precious cells are then sent on a special journey, not through the body, but to a sophisticated laboratory. Here, in a controlled environment, these T cells undergo a remarkable transformation. Genetic engineering techniques are employed to insert a new gene into them. This gene instructs the T cell to produce a chimeric antigen receptor, CAR, on its surface. This CAR is a marvel of biological engineering. It's a synthetic receptor, part antibody, part T-cell receptor, designed to recognize a very specific antigen, a unique molecular signature found on the surface of your particular cancer cells. It's like giving each T-cell a highly specialized molecular lock and key mechanism, where the key is precisely shaped to fit only the lock on the cancer cell, bypassing the usual complex identification process. Once these T-cells are endowed with their new CARs, they are no longer just ordinary T-cells, they are now CAR T-cells, custom-designed warriors, each one a precision instrument of destruction. But their journey isn't over. They are then multiplied, expanded into the millions, creating an army of these highly specialized cancer-targeting cells. Finally, this invigorated, supercharged army is reinfused back into your bloodstream, ready to seek out and destroy the very cancer cells they were engineered to find. It's an incredible testament to our growing understanding of genetics and immunology, a direct partnership with the body's own defenses, but with a highly personalized, engineered upgrade. These CAR T cells don't need the complex dance of antigen-presenting cells or MHC molecules to recognize their target. They can directly bind to the cancer cells and initiate their destructive mission. They are the ultimate guided missiles, precision-engineered to eliminate the threat from within, a beacon of hope in the fight against internal betrayal. Beyond these sophisticated re-engineering feats, we also explore ways to simply teach the immune system more effectively, or to boost its inherent capabilities. This is where therapeutic vaccines come into play. Unlike the preventative vaccines we discussed earlier, designed to shield us from future infections, therapeutic vaccines aim to awaken or enhance an immune response against an existing disease, particularly cancer. Imagine our immune system as an army that hasn't quite recognized its enemy, or perhaps has grown weary in the fight, its memory fading. A therapeutic vaccine, then, is like providing the army with detailed blueprints of the enemy, complete with their unique insignia and vulnerabilities. It presents specific cancer antigens to the immune system in a way that is designed to provoke a strong, targeted T-cell response. It's about training the body's own forces, showing them exactly what to look for, and urging them to mount a more robust, sustained attack. We are, in essence, helping the body to remember, or to learn anew, the identity of its internal adversary, to reignite the fervour of its protective instinct. And sometimes the immune system simply needs a louder alarm, a stronger signal to rally its forces. This is the role of cytokine therapy. We've spoken of cytokines before, those molecular messengers, the alarm bells and navigation beacons of the immune world. In certain contexts, we can administer specific cytokines, like interferons or interleukins, to directly stimulate immune cells, enhancing their activity, encouraging their proliferation, or guiding them to the site of the tumour. It's like amplifying the natural communication network, shouting encouragement to the fighting cells, urging them to redouble their efforts, to coordinate a more powerful, unified front. As we gently traverse these new landscapes of immunotherapy, it's crucial to acknowledge that this journey, like all great scientific endeavours, is not without its complexities and challenges. These powerful therapies, designed to unleash or redirect the immune system, can sometimes lead to an overzealous response. The very concept of releasing the brakes means that occasionally the immune system might struggle to differentiate between the rogue cells and healthy tissues, leading to immune-related adverse events. A form of therapy-induced autoimmunity. It's a delicate tightrope walk, balancing potent efficacy with the imperative of patient safety, a constant negotiation between power and precision. Furthermore, not all cancers respond to immunotherapy, and even those that initially do can sometimes develop resistance. The tumour microenvironment, that complex ecosystem surrounding the cancer cells, can still find ways to suppress the immune attack, or the cancer cells themselves can evolve, shedding the very antigens that the therapy was designed to target. The adversary, you see, is constantly learning, constantly adapting, a testament to the relentless evolutionary pressure of life itself, a reminder that the dance between self and non-self is never truly static. Yet the future gleams with immense promise. Researchers are tirelessly exploring combination therapies, pairing different immunotherapies with each other, or with traditional treatments like chemotherapy and radiation, to achieve synergistic effects. We are delving deeper into the nuances of personalized medicine, striving to identify the specific biomarkers that predict which patients will respond best to which therapy. The development of liquid biopsies to monitor tumor evolution and immune response in real time, and a more profound understanding of the intricate cellular and molecular crosstalk within the tumor microenvironment, are opening new avenues for intervention, new strategies to outwit the cunning enemy. As we come to the close of this chapter, and indeed this profound journey into the heart of immunology, I hope you carry with you a renewed sense of wonder. The immune system, that silent, tireless guardian within each of us, is not just a collection of cells and molecules. It is the living embodiment of our identity, a testament to billions of years of evolutionary wisdom. It defines us, protects us, and in its astonishing complexity, invites us to contemplate the very essence of life itself, and our humble yet growing ability to understand and assist its ancient work. We have moved from the primal whisper of the cell membrane distinguishing self from non-self, through the immediate ancient defences of innate immunity, to the sophisticated, learning intelligence of adaptive immunity. We have seen its triumphs, its vulnerabilities, and now its incredible potential when guided by human ingenuity, a collaboration between our conscious efforts and the body's innate wisdom. To learn about the immune system is to learn about the most fundamental struggle for existence, played out within the microscopic universe of our own bodies. It is a story of constant vigilance, of intricate communication, of life perpetually striving to maintain its boundaries against the ceaseless tide of change and challenge. Thank you for joining me on this peaceful exploration, for lending your quiet curiosity to these profound biological mysteries. As you drift deeper into tranquility, may the silent symphony of your own immune system continue its tireless work, a testament to the astonishing resilience and wisdom woven into the very fabric of your being. Rest now, and let the gentle rhythm of life's most ancient defense lull you into a peaceful, restorative slumber. The journey within continues even as you dream. We have journeyed far, haven't we? From the primal whisper of a single cell defining its boundary, through the intricate dances of innate defense, the sophisticated memory of adaptive immunity, and the delicate balance that sometimes falters. We've explored the profound challenges of autoimmunity, immunodeficiency, and cancer, and peered into the future where human ingenuity seeks to bolster this ancient internal guardian. Now, as our exploration gently draws to a close, let us widen our gaze one last time to see the immune system not as an isolated fortress, but as an integral thread in the vast, shimmering tapestry of our entire being. It is a system deeply intertwined with every breath we take, every thought we think, every morsel we consume. It is a living legacy, constantly evolving, perpetually learning, forever striving for harmony. Have you ever considered the universe within your gut? a bustling metropolis of trillions of microorganisms, bacteria, fungi, viruses, collectively known as your microbiome. And for so long we viewed bacteria primarily as foes, as the non-self that our immune system must eradicate. But the wisdom of life is far more nuanced. This internal ecosystem, a veritable rainforest within your digestive tract, is not just tolerated, it is essential. Imagine it, a vibrant, bustling marketplace where countless species coexist, each playing a role. Our immune system, particularly the vast immunological landscape of the gut, does not simply attack everything that isn't us. No, it has learned a profound lesson in diplomacy and discernment. It recognizes the beneficial members of this microbial community, those scient partners who aid in digestion, produce vital vitamins, and even help educate our immune cells. These tiny inhabitants send out their own gentle signals, a complex language of metabolites and molecular patterns that our immune cells learn to interpret. They are not invaders to be repelled, but rather cohabitants whose presence helps to shape the very fabric of our immune identity. This education begins early, a foundational curriculum for the nascent immune system. Exposure to a diverse range of microbes in infancy helps shape the development of immune tolerance, teaching it what to ignore, what to embrace, and what truly poses a threat. It's like a young sentinel learning the faces of the friendly villagers before being tasked with guarding the gates. Without this early, rich interaction, the immune system can become hypersensitive, prone to misidentifying harmless elements as enemies, contributing to allergies and even autoimmune conditions. The delicate balance of the microbiome, therefore, becomes a mirror reflecting the balance of our immune health. A disrupted marketplace with an imbalance of power among its inhabitants can send ripples of discord throughout the entire immune symphony. But the immune system's interconnectedness extends beyond the microscopic world within us. It is profoundly influenced by the macroscopic world we inhabit, and even by our internal landscape of thoughts and feelings. The ancient wisdom embedded within our cells understands that self is not merely a collection of molecules, but a holistic experience. Consider the gentle rhythm of your breath as you listen, the subtle rise and fall. How does the hurried pace of modern life, the relentless drumbeat of stress, impact this silent guardian? When we experience chronic stress, our bodies release hormones like cortisol, powerful messengers that, in short bursts, can be helpful. But prolonged exposure can subtly suppress certain immune functions, dampening the vigilance of some cells, altering the delicate balance of cytokines. It's as if the constant alarm bells of stress make it harder for the immune system to hear the true threats, leaving it distracted and less effective. And what of sleep, that nightly descent into the quiet depths of restoration? During these hours of repose, our immune system is far from dormant. It is busy repairing, consolidating immunological memory, and producing crucial protective molecules. It's a time of quiet industry, where the day's battles are analysed and future strategies are refined. A consistent lack of sleep, that relentless erosion of our resting hours, can impair the immune system's ability to mount effective defences, making us more susceptible to infections and potentially hindering its surveillance against rogue cells. Sleep is not merely a luxury, it is a fundamental pillar of immune resilience. A time when the body's inner mechanisms recalibrate and strengthen. Even the food we choose to nourish ourselves with, the very building blocks of our being, plays a profound role. A diet rich in diverse, whole foods provides the essential nutrients, the vitamins, minerals, and antioxidants that fuel immune cells and support their complex functions. Conversely, diets lacking in these vital components, or those dominated by inflammatory agents, can create a subtle, chronic hum of distress within the body, diverting immune resources and potentially contributing to systemic inflammation. Our plate, in a very real sense, becomes a part of our immune strategy, either supporting its harmony or introducing dissonance. This understanding, this recognition of the immune system's deep roots within our overall well-being, brings us to a crucial realization. We are not passive observers of our inner universe. Through conscious choices, through the cultivation of calm and balance, we can actively support this remarkable system. We can nurture its strength, honor its wisdom, and contribute to its harmonious functioning. Yet even with all our understanding, all our efforts, the immune system remains in a constant, timeless dance with the world beyond our skin. The pathogens it battles are not static foes. They are evolving entities, constantly adapting, finding new ways to evade detection, to mimic self, to exploit vulnerabilities. This ceaseless evolutionary arms race playing out over millennia has sculpted the astonishing complexity and adaptability we have witnessed. Our immune system is a living testament to this ongoing dialogue, a dynamic entity shaped by the very challenges it overcomes. New viruses emerge, bacteria develop resistance, and even our own cellular invaders, cancer, find novel strategies to escape surveillance. This means the quest for understanding, for new therapies, for deeper insights, is an unending one. The scientific journey continues, fueled by curiosity and a profound respect for the intricacies of life. Researchers around the globe are still unraveling the mysteries of long-term immune memory, exploring the full potential of the microbiome, designing ever more precise immunotherapies, and seeking to understand the subtle interplay between our minds, our bodies, and our defenses. The story of immunology is one of continuous discovery, a testament to the boundless complexity of life itself. As we bring this particular journey to a gentle close, let us carry with us a renewed sense of wonder for the silent symphony within. Think of the billions of cells working in concert, each a tiny sentinel, each a dedicated artisan, building and maintaining the fortress of your identity. Feel the quiet hum of their activity, the constant vigilance, the tireless dedication. This immune system, this living embodiment of self, is a gift. It is the legacy of billions of years of evolution, a testament to life's unwavering will to persist, to adapt, to thrive. It is a reminder of the astonishing complexity that allows us to experience the world, to learn, to love, to simply be. So as the gentle currents of these words carry you deeper into tranquility, perhaps you can feel a profound connection to this inner universe. A connection not just of scientific understanding, but of deep appreciation. For the immune system is more than a biological mechanism. It is a philosophy etched in flesh and blood, a constant affirmation of I am in the face of all that is not I. Thank you for joining me on this exploration, for lending your quiet curiosity to the wonders within. May this newfound understanding bring you a deeper sense of peace, a profound appreciation for the magnificent life force that resides within you. Rest now, knowing that your inner guardians are ever vigilant, ever learning, ever preserving the precious harmony that is you. Sleep well and dream of the extraordinary universe you carry within. We have journeyed far, haven't we? Through the vibrant landscapes of the innate and adaptive, across the battlefields of autoimmunity and the quiet resilience against cancer. We've touched upon the ancient wisdom encoded in every cell, the tireless dance of defense that safeguards the very essence of you. Now, as the horizon of our understanding expands, we turn our gaze toward the future, toward the echoes of tomorrow, where the silent symphony of immunity promises even grander revelations. Consider, if you will, the vast uncharted territories that still lie before us. While we have peeled back many layers of this intricate tapestry, the threads of its deepest patterns remain somewhat elusive. We speak of personalized immunotherapies, a concept as revolutionary as it is complex. Imagine a future where your immune system, with its unique history and genetic blueprint, is not merely understood in broad strokes, but in exquisitely fine detail. We are moving beyond the general map to a highly specific molecular GPS, guiding interventions tailored precisely to your individual needs. This isn't merely a dream, it is an unfolding reality, propelled by the relentless curiosity of human endeavour and the astonishing power of technology. Think of artificial intelligence not as a cold calculating machine, but as a tireless, discerning librarian sifting through mountains of genomic data, proteomic profiles and clinical outcomes. AI can discern subtle patterns that escape the human eye, predict how a particular cancer might respond to a specific checkpoint inhibitor, or identify the precise mutation that renders an immune cell less effective. It's like having a master conductor who can not only hear every instrument in the orchestra, but also anticipate every note, every nuance, creating a perfect harmony of treatment. We are beginning to unlock the full potential of precision immunology, where therapies are designed not just for a disease, but for your disease, in your body at this moment. This involves sophisticated diagnostics that can map the unique landscape of your tumor's immune microenvironment, identifying the specific immune cells present, their activation states, and the exact molecular signals they are exchanging. It's like reading the intricate individual story of your body's struggle, then writing a new chapter of triumph. Yet, with every answer, new questions blossom like delicate, luminous flowers in the scientific garden. How do we fully harness the power of the microbiome, that bustling inner city of microbes, to further educate and fine-tune our immune responses? We know it plays a profound role, a silent co-conspirator in shaping our immune identity from birth. But the precise language it speaks to our immune cells, the subtle dialect of metabolites and microbial products, is still being translated. Could we one day cultivate a bespoke microbial ecosystem within you, one designed to optimize your immune system's vigilance against disease or to temper its overzealous reactions in autoimmunity? The potential is immense, suggesting a future where our inner microbial gardens are tended with the same care as our physical health. And what of the enigmatic realm of immune memory? We've explored how B and T cells carry the indelible imprints of past encounters, standing ready for future invasions. But the nuances of this memory, its longevity, its specificity, how it wanes or strengthens over time, are still subjects of intense investigation. How do memory cells persist for decades, sometimes a lifetime, without constant restimulation? What are the molecular archives that hold these vital blueprints, allowing for such rapid, potent recall? It's as if our cells possess an ancient library, constantly updated, yet largely operating beneath the surface of our conscious awareness. Understanding this deeper architecture of memory could unlock new strategies for more effective, long-lasting vaccines, or even for retraining a faulty immune memory that contributes to chronic conditions. Then there is the profound interplay between the immune system and our nervous system, the intricate dialogue between mind and body. We are only just beginning to grasp the full extent of this bidirectional communication. Stress, as we know, can profoundly impact immunity, but the exact molecular pathways, the neural circuits that translate emotion into immune modulation, are still being mapped. Could we learn to consciously influence our immune system, perhaps through mindfulness or specific mental practices, to enhance its protective capabilities or calm its inflammatory responses? It's a fascinating frontier, blurring the lines between what we perceive as purely physical and what is profoundly mental, suggesting an even deeper unity within us. As we stand at the precipice of these future discoveries, it's important to remember the fundamental questions that initiated our journey. What is me, and what is not me? The immune system, in its tireless discrimination, is the living embodiment of this distinction, the biological philosopher within. It constantly monitors, assesses, and adapts, maintaining the delicate equilibrium that allows you to exist as a coherent, unique entity. Have you ever considered the sheer elegance of this system? The way it has evolved over billions of years, from the simple membrane-bound distinctions of the earliest cells to the astonishing complexity of adaptive immunity. It is a testament to the power of natural selection, a grand, ongoing experiment in survival and adaptation. Each time a pathogen mutates, each time a cancer cell tries to evade detection, the immune system learns, evolves, and finds new ways to respond. It is a dynamic, ever-changing entity, never static, always in motion, much like life itself. And this brings us back to the core of our exploration, the profound wonder of the human body. The immune system is not just a collection of cells and molecules. It is a narrative, a story of vigilance, sacrifice, and an unwavering commitment to life. It is the silent guardian, the unsung hero performing its intricate ballet within you, day in and day out, without your conscious command. In this quiet space, as the world outside softens and the edges of your awareness blur, I invite you to simply feel this profound presence within. Feel the sonant strength, the ancient wisdom, the continuous protection. There is a deep comfort in knowing that this intricate, tireless system is at work, constantly maintaining your integrity, protecting your individuality. It is a privilege, isn't it, to glimpse into this hidden universe, to understand even a little the astonishing complexity that allows us to thrive, to dream, to experience the world. This journey through immunology is not just about scientific facts. It is about appreciating the miracle of life itself, the delicate flame that is constantly tended by an invisible internal fire. As we prepare to drift into deeper repose, carry with you this sense of wonder, this gratitude for the unseen symphony playing within. Let the knowledge we've shared become a gentle lullaby, a soft whisper of understanding that settles deep within your consciousness. The immune system in all its magnificence is a constant reminder of the incredible resilience and interconnectedness of life. Thank you for joining me on this exploration, for lending your quiet curiosity to the wonders of medical science. May this newfound appreciation for your body's silent guardians bring you a profound sense of peace. Rest now, knowing that within you, an ancient, wise, and astonishingly complex system stands ever vigilant, a testament to the enduring miracle of you. Sleep deeply, and may your dreams be filled with the quiet confidence of a body wonderfully made, protected by its own magnificent, unseen symphony. We have journeyed together through the intricate pathways and silent sentinels of your inner world, exploring the profound wisdom etched into the very fabric of your being. Think of these pathways as ancient labyrinthine passages winding through the core of your existence, each turn revealing a new facet of an astonishing self-regulating universe. And the sentinels? They are the ever-watchful guardians, standing guard at invisible thresholds, their vigilance a constant hum beneath the surface of your conscious awareness. From the primal whisper of self and non-self, that fundamental existential query echoing from the dawn of multicellular life, to the sophisticated ballets of adaptive immunity, where cells perform intricate choreographed defenses with breathtaking precision, we've witnessed a miracle unfolding within you. It is a timeless story of vigilance and resilience, written in the very language of life itself, a narrative far more complex and compelling than any epic tale. But the narrative of immunology, like life itself, is ever unfolding, a boundless frontier where every answer begets a new, more profound question, pulling us deeper into its enigmatic embrace. As we drift deeper into the quiet contemplation of this astonishing system, allow your mind to expand, to truly consider the vast, shimmering expanse of what we still seek to understand. These are the mysteries that beckon us forward, not with a clamour, but with a gentle, insistent pull, promising revelations that could reshape our very understanding of existence. The frontiers of immunology are not just sterile laboratories and powerful microscopes, though these are vital tools. They are, more fundamentally, the very edges of our comprehension. They challenge our assumptions, pushing us to redefine health, identity, and the intricate, delicate dance of life itself. It's a journey into the unknown, guided by the light of scientific inquiry and the profound wonder of discovery. Imagine, if you will, a future sculpted by the very patterns we've begun to discern within this magnificent internal architecture. We spoke of artificial intelligence, and how it might one day serve as a tireless cartographer, an indefatigable explorer mapping the uncharted territories of your immune landscape with a precision previously unimaginable. Picture, in your mind's eye, algorithms so vast and intricate they resemble a galactic nebulae, shimmering with countless points of data, each a star in your personal biological sky. These digital intellects would sift through oceans of cellular information, the silent communications between lymphocytes, the energy signatures of macrophages, the subtle genetic expressions of every immune cell, not just observing, but predicting. They would anticipate the whispers of disease long before they coalesce into a roar, identifying the minute, almost imperceptible shifts in your defenses that signal an impending challenge. This isn't merely the stuff of science fiction, it is the burgeoning reality of personalized immunology. In this future, your unique immunological fingerprint, the intricate sum of your genes, the diverse ecosystems of your microbiome, and the cumulative tapestry of your life experiences becomes the blueprint for a bespoke suit of armor. This isn't a generic mass-produced defense, but one tailored precisely to your individual needs, woven from the very fabric of your being. Think of the profound relief, the deep sense of security, knowing that your cellular guardians are not just reacting, but proactively guided by unparalleled precision, a hyper-intelligent conductor leading a symphony specifically composed and performed for you. It's a shift from broad-spectrum intervention to exquisite individualized care, transforming the very paradigm of health from reactive treatment to proactive, deeply personal optimization. And what of our inner gardens, These sprawling, diverse, and often hidden ecosystems we call the microbiome. Imagine them as lush, vibrant rainforests within you, teeming with billions of microscopic life forms, each playing a crucial role in the grand orchestra of your internal self. We've touched upon its profound influence, its ancient pact with your immune cells, a silent millennia-old agreement forged through coevolution, where these tiny inhabitants educate your immune system, shaping its responses, teaching it what to tolerate and what to defend against. Yet despite this deep history, we are only beginning to grasp the full, astonishing extent of this symbiotic relationship. What if in the not-so-distant future we learn to cultivate these inner universes with such finesse, such deliberate artistry, that we could prevent devastating autoimmune diseases like rheumatoid arthritis or Crohn's before they even take root, gently guiding the immune system away from self-attack? Or imagine bolstering your defenses against novel pathogens, viruses and bacteria yet unknown, simply by nurturing the right microbial communities, creating an unassailable fortress from within. The prospect extends even further, modulating moods, sharpening cognitive function, and enhancing mental clarity by carefully tending to these microscopic allies. It is a truly fascinating vision, cultivating health not just by treating symptoms after they arise, but by harmonizing the fundamental microbial conversations that underpin your very existence, fostering a deep, resonant well-being that emanates from your core. This isn't just about gut health, it's about a profound re-engineering of vitality, a silent revolution in personal well-being. Then there is the whispered conversation, the profound, intimate dialogue between the immune system and the nervous system, two halves of a single conscious whole, once mistakenly thought to be separate sovereign kingdoms. We are now learning that the brain, that magnificent organ, the very seat of your thoughts, emotions and consciousness, is in constant dynamic dialogue with your immune cells. Imagine it as a continuous, silent exchange of vital information, a highly sophisticated internal network. Neurotransmitters, those swift chemical messengers of your mind, serotonin, dopamine, cortisol, can directly influence immune responses, acting like urgent dispatches sent from the command center. And in an astonishing reciprocal loop, immune signals like cytokines can traverse the blood-brain barrier, affecting brain function, shaping everything from your mood, your focus, your memory, even your very perception of reality. Have you ever truly considered the profound implications of this connection? To think that your stress, that gnawing anxiety over a deadline, or your boundless joy from a cherished moment, your deepest reflections on life's meaning, might be directly shaping the vigilance and responsiveness of your cellular guardians, your internal army. It's a humbling and empowering thought. This understanding suggests a truly holistic paradigm of health, one where mental well-being is not merely a pleasant or desirable state, but a fundamental, non-negotiable pillar of immunological strength. It means caring for your mind is caring for your body, and vice versa, in the most literal sense. Understanding this intricate dialogue, mapping its myriad pathways, could unlock entirely new avenues for treating intractable neurological disorders, debilitating chronic pain and pervasive mental health challenges, finally weaving together the magnificent tapestry of mind and body in ways we are only just beginning to comprehend, revealing an interconnectedness more profound than we ever dared to imagine. This continuous evolution, this timeless ballet between life and its myriad challenges, is another profound aspect of the immune system story, a saga written across eons. For millions of years, stretching back to the earliest multicellular organisms, your ancestors' immune systems have been locked in an intricate high-stakes arms race with pathogens. Imagine this relentless dance. A pathogen develops a new stealth mechanism, and within generations your immune system counters with an innovative detection strategy. Each viral mutation, each bacterial resistance has prompted a counter-evolution in our defenses, a testament to the relentless ingenuity and creative force of life itself. It is a dynamic equilibrium, constantly shifting, perpetually refining. The immune system you possess today is not merely a modern marvel, it is the breathtaking culmination of this vast ancient history. It is a living record, inscribed in your very DNA and cellular memory, of countless battles won, lessons painstakingly learned, and hard-earned wisdom passed down through generations. It carries the echoes of struggles against ancient plagues and forgotten infections, a biological library of survival. This is a system designed not for stasis, not for a predictable, unchanging world, but for dynamic adaptation, an ongoing, intelligent dialogue with a perpetually changing environment. And as new threats emerge, from insidious environmental toxins to rapidly evolving novel viruses, challenges our ancestors could never have conceived, your immune system stands ready. It draws upon this deep ancient wisdom, yet simultaneously taps into its astonishing capacity for innovation, ready to write the next chapter in its endless story of defense and resilience. But perhaps the most profound mystery, the one that resonates deep within the core of our being, touching upon the very essence of existence, is the elusive concept of self. We began our journey with this primal distinction, the fundamental echoing question that has plagued philosophers and scientists alike. What is me, and what is not me? Your immune system, in its unparalleled wisdom, acts as the ultimate arbiter of this identity, a living, breathing, constantly re-evaluating boundary that defines your physical self. Imagine it as an ever-vigilant border patrol, tirelessly patrolling every cell, every tissue, questioning, identifying and protecting this sacred personal boundary from any perceived intruder. It's a silent, ceaseless interrogation. Are you friend or foe? Are you part of me or an unwelcome guest? Yet this definition is not static, not a rigid line drawn in the sand. It is fluid, shifting with age, adapting with every new experience, and even evolving with the very cells that constitute your body, which are constantly being renewed and replaced. Consider the profound implications. What does it truly mean to be you when your skin cells shed, when your blood cells are replaced every few months, when your microbiome shifts its composition, when your thoughts and experiences continually reshape your neural networks? The immune system, in its unwavering, vigilant defense of self, offers not just a biological mechanism, but a profound, tangible lens through which to ponder one of life's greatest philosophical questions. It reminds us with every heartbeat that identity is not a fixed point, a singular, unchanging essence, but rather a dynamic, resilient, and constantly negotiated state of being. It is a testament to the beautiful, intricate impermanence that defines life itself, a continuous act of becoming. As we gently bring our exploration to a close, let this profound understanding settle deeply within you, permeating,